Hello, everyone, and welcome to today's webinar on particle characterization. My name is Rena Samsu, marketing at EAG Laboratories, a Eurofins company. Before we get started, I'd like to go over a few housekeeping items for today's event. All attendees have been muted. However, we'd still love to hear from you during today's presentation. You will have the opportunity to submit text questions to today's presenter by typing your questions into the questions panel located in the bottom right of your screen. You may send in your questions at any time during the presentation. We will collect these and address them during the Q&A session at the end of today's presentation. At the end of the webinar, a survey will pop up on your screen. Your feedback is greatly appreciated and will help us to improve our future events. I would now like to introduce Lynn Zhang, Senior Technical Specialist at EAG Laboratories.
Hi, Rena. Thank you. Welcome, everybody. Thank you for joining me today for the webinar of particle characterization. We're going to focus on analyzing and reducing particulate matter. Before we start, I will give a little bit introduction of myself. My grad school group was the LCMS group. My work particularly is more focused on optimizing mass spec conditions rather than the separation. As I was a mass spectrometrist by training, in general, we need to get things in solution first. One part I had to learn quickly when I joined EAG was to analyze as is for those techniques. For a lot of cases, we have studies like requires a minimum amount of sample prep, and it has been I have been with EAG for 5 years now, and there certainly has been a lot of practices on those analyze as techniques, especially for particle characterization. Today, I'll share some of my experiences with you. Today's webinar will include several different parts. Of course, we started with a little introduction of EAG, then we'll go through the smart chart and talking about something about particulate problems and particulate analysis, and of course, the reduction of particles. And at the end, we'll have a little summary. So talk a little bit about EAG. EAG has several hundred of highly educated scientists and engineers working in laboratories around the world. Our range of equipment and diversities of expertise allows us to offer very comprehensive material analysis, serving many different industries. We provide peer review reports, often within a few days of sample reception, and when necessary, we can even provide same-day turnaround when a quick resolution is critical for our clients. We have about several different laboratories, of course. The majority of them are in North America. However, we do have a few laboratories in Asia and Europe. As of now, all EAG laboratories around the world are currently open to serve our clients at this difficult time. Here is our smart chart. So you can see here the x-axis are the spot sizes, and the y-axis are the detection ranges of concentration and the corresponding number of atoms. And the bottom are the imaging techniques in such like SEM, TEM. And the red side here, we can see the Bock techniques we use in the lab. They provide information more or less representing the entire sample if the samples are homogeneous. Of course, my favorite one here is the LCMS. When we talk about particles, there are two kinds of particles in general. One are the ones we like, and the others are the ones we don't like. And the ones we like are the bulk materials. So the powdered samples that serves as raw material of your production or finished goods. For these, we want information about the powders. For example, we want particle size and distribution. We want to know the particle shape. What do they look like shape-wise? And if they have a coating, what's the coating thickness? Is there any aggregation happening with the particles? And some more information as well. And some examples of this kind of powder materials, like, for example, titanium oxide is fairly often used in industries, and nanoparticles as well. The other part of the particles are the ones we don't like. They're generally contaminations. We also want to gather information about them as well. We want to know the particle size and how many particles are there, and the general morphology of it, of course, chemical identity as well. Once we have all this information, we want to figure out where the source of these particulates are and potentially remove them as preferred. And some of the examples that we usually call them foreign materials, foreign particles, fibers are fairly common with the ones we see. And today we have the focus is on the foreign particles. We'll discuss it a little bit more. When you say there's a foreign particles, why do we care? Of course, in different industries have different reasons why they don't like particulates in their sample. There's some examples, like for example, in the medical device and pharmaceutical industry, the potential risk to patients and are the ones we may have main concern in those industries. And of course, other industries, including medical device, and the functional effect. You don't want the malfunction of your parts that could lead from the particle contamination. And even just a cosmetic damage or defect, we don't want them either. And when we talk about particle analysis, why they're there is because they're really everywhere. They're in the air. They're part of our body, like our hair. Our dead skin could contribute to that as well, and sand. And different industries all have like different requirements on how many particles we need to be there and sizes. A general like a guideline for the medical device industry, really depending on the device. But the general guideline is for particles 25 microns or larger, they're no more than 600. For particles more than larger than 10 microns, no more than 6,000 is there. And all the particles observed chemistry of the particle is preferred to be known. So by that I'm saying is to understand what's the chemical composition of the particle that would be used for potential toxicology review to figure out that long-term effect or short-term effect. In general, when we talk about particle characterization, there are several things involved in that. Like I said, sizing, mythology, and the chemistry of the particles. For sizing, the medical device and pharma industry have their clear guidelines. The USP 780 and 789 describes the method to do the sizing. And 787 are relatively newer. They're good backup if 788 cannot be used for your sample. And of course, other industries have their guidelines as well. Aerospace has their own, and that method has been highly adopted by other industries to use. Morphology-wise, usually requires some sort of imaging technique. So optical microscopes and SEM are commonly used, and virtual inspection as well. Like human eye is a very powerful detector. The chemistry of the particles and usually done by some type of analytical instrument. Commonly used ones like FTIR microscope and Raman, SEM-EDX, and there are other techniques available as well. We'll go into these three parts in a little bit more details later. So talk about particle sizing and counting. Their 788 method describes two different methods actually in it. The methods are called like analyzing particulates in injection and parental infusions. The first method is a light obscuration method, and the method is described as method 1. It's highly automatic method. How that works is you have particles in solution by comparing that solution to a standard solution. So the standard solution will contain 10 micron particles and no amount of 25 micron particles as well. By comparison, you can tell the size, the distribution of the size, and also the number of particles, the particle density as well. The second method, we call it microscopic method, which is a similar solution with the particles in it going through a filter. There's an example of the 788 filter. It's a membrane filter, so it goes through this filter, and the solution will filter through, and the particles will be captured on the surface of the filter. So you can see the filter has some little dots on it on the side and different color as well. So the larger one is the 25 micron and the smaller one, the 10 micron for comparison with the particles directly. And you can see a linear scale ruler as well. So these are, they all have their benefit and downside as well. So light obscuration is highly automated. You have the solution, you get a particle size and distribution. You're good. And microscope method is time consuming. Of course, you have to manually filter in and counting the particles and their eyepiece of the optical microscope. But the benefit is you have direct access of the particle. You already separate them from the solution. You have a particle on the filters. Once we've had the particle collected, we talk about the particulate morphology. How do we categorize them? What do we describe the particles we see? To do that, everyone said we need some kind of imaging technique, a microscope, and a very good chemist, like experience with particulates, would describe them the way as to common sense and to particular categories they belong to. So when you talk about mythology, we go through this little chart. Like I said, it's more common sense. You have general appearance. Does the particle look like a fiber? Is it a crystalline, or is it globular? These are just general description of it. And of course, the optical of the particle. What color is it? Is it white? Is it red? What kind of color is it? Is it transparent? Semi-transparent? No transparency at all? What's the shape of the particle? Is it sphere? All of these are contribute to the particle description. One last one is a very good character is the surface roughness. If you say the surface smooth, is it porous? Is it sharp edged? So these are all the terms we use to describe a particle. Now let me go through a practice. If I describe a particle using the morphologies we just discussed, let's see what you can picture that particle. If I say a particle looks like a strip, but a little bit globular, it's silver color, shining on the surface, sharp on the edge. Can you picture what it is? So that's the picture, that's the particle I just described. It's a metal shave. We actually see it fairly often for particular analysis. So it may contribute from a machine, have a metal shave, or sometimes especially the pharmaceutical industry have metal caps. We see a lot of aluminum caps may have aluminum shave still attached to it, and sometimes in the period they'll fall into the bottle. So once we have the morphology determined, we want to get chemical information. We're talking about the chemical characterization. But before I go into the details on the instrumentation, I want to talk a little bit about the isolation of the particles. The reason is like for 788, if it's using the filtration and macroscopic method, we have direct access to particles. But it's not always the case for particular analysis. Sometimes we may get single particles coming from a inspection booth during our production line, or sometimes on the finished device, or a part that has the particulates attached to it. Then we'll require isolation to gain access to that particulate. So when we're talking about isolation of particles, there usually is two parts. When the particle is in the liquid solution, or the particle is on another solid sample. When it's in solution, it goes through several different steps. I have an example here. So this is a bottle of a drug product, and some kind of aqueous-based buffer, and I have a little red fabric-looking particle there. Usually the process is we can either fish this particle out or go through a decant and filter the small amount of sample containing the fiber particle, and then rinse the fabric particle and then dry it for the future analysis. And if it's a particle on the solid sample, then we normally do is to determine is the particle on the surface, loosely attached to the surface, or embedded in. The approaches for these are different. If it's on the surface, if we can just isolate taking the particle out, that would be great. If it's slightly embedded, you can visually see but don't have access to it, those are the trickier ones. There requires a comprehensive sample prep to obtain the particle. So usually when we actually finally analyzing the particle, we take it out. We'll do is to analyze the unknown particle and also the solid particle, the solid material around it as a control. If we can remove the matrix, that would be great. If we can't, we at least know which signal is contributing from the matrix. So isolating of particles, matrix removal is the key. Okay, so now we have particles isolated. We'll go through analytical considerations. How do we go into the chemical characterization? Which techniques to choose and why? There's a lot of questions we ask ourselves. We look at the particle. Is this particle homogeneous? If it is homogeneous, yes, we can take one big spot and perform the analysis, and that will be the information we're requiring. If it's not homogeneous, if it requires a general observation of several different parts, you can see this is a different color with the other end, and the morphology of one particle is different than the other side. Then you want to analyze the different parts of the particles, make sure you get as much information as possible. And of course, the size of particle, how large it is. The larger they are, we have more samples to work with. And if it's homogeneous, we can split one particle to two, go into two different techniques. But then if it's really small, that's usually where the challenge is. When it's really small, you have to pick and choose which analyte to do first. You don't want the first technique you picked consumed your sample and didn't give you enough useful information, and you already lost your particle. That's the last thing we want to see. And some other questions you ask, is this particles organic or inorganic? These are usually can be a direct observation from the morphology when we look at it. May not necessarily be true always, but it give you a good starting point of which techniques to try first. Is the sample vacuum compatible? That's a very good question you want to consider first, because some techniques requires a high vacuum chamber. You don't want to put your samples in that's not compatible. I may have waters or liquid trapped inside it, and it exploded in the instrument when it hits high vacuum. And not only you lose your particle, you may very likely contaminate your instrument as well. And of course, how thick the particle is. Some of the techniques only penetrate a small thin layer of it. If we want bulk material, do we want to have a cross section? Is the particulates homogeneous throughout the different layers? Those are the questions we ask as well. Here are the two boxes of EAG. So we have all different techniques, and we talked about earlier, our smart chart gives the techniques we have. We actually have beyond just this chart, we have some other techniques as well. You're more than welcome to go to our website. The smart chart is on our website, and it's interactive. Some techniques you're interested in, just click on the bubble, and that will bring you to the technique page. You can read it, and associate some white papers and the case studies we have as well. All right, so let's go into the two primary techniques we use for particulate analysis. That will be FTIR and SEM-EDX. So FTIR is used to determine major chemical family and functional groups. And the EDX technique is to determine the major elemental profiles. We'll go into them one by one. First, let's talk about FTIR a little bit. FTIR is located on the right side corner of our smart chart. It's highlighted here. And the bottom is, like I said, is the analytical spot size. So it goes to about double digits micron all the way to a little over 1 millimeter. And the concentration is for bulk material. So basically we can go from 100% pure as low as we can push to about 100 ppm. It really depends. Sometimes some materials has a very strong IR band. We may even be able to push a little bit lower than that. But the end is it's really fair to say is FTIR is the best technique to identify unknown organic materials. The organic is the key part here. Like we discussed earlier, FTIR provides molecular information. We can get a functional group and chemical family. And how that works is IR radiation is absorbed by the sample to produce a molecular vibration, stretching, or to add the frequency that's unique to a certain organic chemical bond. And because the different chemical bonds have different signatures, and all of them together and lead to a different chemical structure, will have a different fingerprint, the spectral fingerprint. From the fingerprint, we can relate back to what kind of chemical they are. EAG has an extensive library databases. We can have a great match. For particular analysis, commonly used is a microscope FTIR that provides a ability to analyze very small samples. So a microscope FTIR basically is a microscope coupled with FTIR. The name speaks for itself. There's 3 general measurements for particulate matter analysis: ATR mode, transmission mode, and reflectance mode. For particular analysis to analyze is minimum amount of sample tempering. My favorite one is the microscope ATR mode. As you can see here, is more like a general setup. So the sample is placed on the ATR crystal, and the IR beam is coming from the bottom of it. So the majority of the signal coming from is the interface between the two. So the sample basically is on the bottom surface of it. It's great for powder, plastics, and solids, and paints, and coatings. Like I said, it's minimum amount of sample prep is required, and you analyze a sample in its nature state. And of course, the transmission and reflectance has its benefit as well. I'm not going to too much details on this part. Later on, we'll have a webinar about the FTIR and Raman. You guys are more than welcome to join. We'll go into more details on those techniques and different sample prep and sampling mode. Once, it doesn't matter which technique, which sampling mode you use on the FTIR. At the end of the day, you get an FTIR spectrum. Now, interpreting this spectra is very important. This is a spectra like only give you what the fingerprint look like. How you use that fingerprint to get the data you want is very important. Of course, step number 1 is when you have a spectra, you always want to search the library to see what's the top library matches. If the computer can do your work, definitely let them do that. It's more efficient. You get your information right away. So you search the top library, you get a match. A match is great, that's good. If you don't have a good match and the spectra is complex, or what do you do next is you want to search a specific area in the functional group region. So if you search 1 functional group or 2 functional groups, see what other compounds have these functional groups in common. And recognize common materials. For example, like PDMS, we see fairly often as a background or by itself. So PDMS is, our makeups and like hair gels. It's everywhere around us in production as well. Calcium carbonate is sometimes as a rubber filler. We see them fairly often as well. Talc. And proteins and cellulose is a lot of the fiber materials are cellulose fibers. And silica is a fancy word for sand. So we see them fairly often as particulates. And you realize these common materials not only because you can see them when you see the spectra. Some of them are fairly simple. I'll show you a few in a little bit. Some of them are fairly simple, but it's very important is when these simple materials, the common materials, as part of your complex spectrum, serves as because your particle is a mixture of these things with something else, some more complex organic compounds. So if you isolate these common materials, they may give you a better match of the other side, the other part of the particulate is. And of course, you can also use do the old school way to use the absorption bands to corresponding to their functional group to figure out what's the basic functional group is, and based on shifting, you can like solve a puzzle basically. For example, if you see something around 1700, 1800, that's most of the times is a carboxylic group, a carbonyl group. So if as you see our carbonyl group, and the different shiftings could be related to what kind of functional groups they are attached to, lead to this kind of shiftings. We do have a long list of these kind of compounds. These are just very, very basic, commonly used ones. Our FTIR chemistry in lab, their books could be like pages and pages long of these kind of tables. We all have our little cheat sheet taped inside our cubicles. This one is my favorite one because sometimes you forget about, you look at it, the basic ones, and they all have shiftings. We'll go into that in more details, and it's really case by case study. When you have a project, contact us. We'll help you find out what they are. Like I promised earlier, we'll have some common materials to show you the spectra. You can see here, this one's a PDMS. Very distinct bands, and they're very intense on those peaks as well for their functional group. And this is a calcium carbonate. We see this one as a base fairly often because, for example, if you have some sort of rubber material, then you get to this blob at the bottom, like as a background, and some other functional groups on top of it. Sometimes they give you a little bit of information that calcium carbonate could be used as a filler. This one is a silica. Okay, so these are very simple spectrums. We see them, we know what they are. If you have a very busy spectrum, then what do you do? And this one is a basic spectrum. I just use it as an example, but it's not a complex spectrum. This one is coming from my database, caffeinic acid. If you search our database, you get one that's 100% 100% match. So what we normally do is you can do a library search if it's not going to match. You can search like several different functional groups. I like to search the region. We call it functional group region. Is the ones about 1800 to 1000 wave number somewhere there. These are characteristic peaks usually. Just a fun fact to throw there: caffeinic acid has almost nothing to do with coffee. Why do I say almost? Because it's actually a key intermediate in the biosynthesis of lignin. So lignin is found in all plants. So does caffeinic acid, including coffee plants. So almost nothing to do with coffee. All right. So now we have analysis going through already. So how do we report them once you have a match? So if you have a great match, then how we normally report it is as this is the unknown particulate is consistent with blah blah, whatever the chemical is, the match. And we do is to have a stacked spectrum of your sample and the matching spectrum to show they are a good match. And if there's no good match or coming from a direct search, we'll search a certain functional region, functional groups, and the chemical family and suggest that these are similar to certain compounds. Or if there's nothing even that close to match your sample, we'll do is to break down the functional groups and chemical families to suggest these are the things as part of your structure. And of course, if it's a common material or in EHG internal library, we would say our experience suggests that this is a possible ID. We could match it if you provide reference sender or resourcing reference sender to compare it to. So just like any other techniques, FTR is great for bulk material ID for organic materials, but it has its limitations as well. The only materials that absorbs radiation are in the about 7,500 to about 400 wave number. That range will provide a good FTIR spectrum. And with the microscope method, that range will be narrower even. It's about 4,000 to 550 wave number, somewhere there. The analytical area of a micro FTIR at EHG can go up to 0.5 millimeter to about a couple millimeters, somewhere there. It's a limit of the microscope. You can't change, push it to be bigger. But if we do a bench ATR without the microscope, we can push it to like 4 or 5 millimeters, a larger area. The analytical area of a microscope ATR can be pushed down to very, very low. Generally, we can say like 10 to 30 micron is a rule of thumb. I like to call it double digits. Once you have a double digits micrometer size particulates, we will be able to get some FTIR information. Now, like I said, if something has strong error band, we can push it to single wave number as well. Sorry, the single microns as well. But that's very challenging to do. After FTIR, we can talk a little bit about EDX. So SEM EDX provides elemental information. Some people like to call it EDS. The S stands for spectroscopy. Some people like to call it EDX for X-ray. They are the same thing, just a preference. So SEM EDX is actually located in the middle of our smart chart. The analytical spot size can go to the sub-micron all the way to about 100 micron. And the intensity, the concentration, as long as it's above like half %, we should be able to get a good elemental information. The thing about SEM EDX is SEM itself is an excellent technique for obtain high resolution images. Of course, EDX is what we want to get elemental content, elemental information. But there you get a bonus of a nice pretty image. We'll show you some later. How SEM works is that there's an E-beam generates the electrons as an energy source, basically, get focused by a couple of different lenses and to hit on the surface of the sample. And the energy will lead to excited or ionize the sample and lead to electrons, different electrons coming off the sample. So there's 3 different detectors to detect the different radiation or different emission from the sample. Of course, we have the EDX detector and the backscattering detector along with the secondary ion detector. This is a typical SEM setup. So the ones we focus on is the EDX detector. That's our friend here. How EDX and we get the X-ray, how it works is that I'm not going into too much details. I'm going to fly through these. So we basically have a excited electron, basically when the E-beams come in, and you get an excited ion because a secondary ion is removed in the layer. And the relaxation from an outer layer electron going in is the one lead to the X-ray fluorescence. That's a signal we're detecting that lead to our elemental information. Like I promised you, we're going to show some pretty images of SEM. Regardless of what EDS image you're doing is, then you get some good pictures as a benefit, as a plus. So this one is actually table salt. The EDS we see will be sodium and chloride, but you get a beautiful image and the nice cubical crystal looking salt. And we get sphere particles as well. At EAG, we do have programs that can do particle counting and particle size distribution analysis through SEM images. So you can see here is a different color associated with a different diameter of the particle, and we can provide distribution with that. That's our algorithm we wrote to provide that information. The smart chart, the smart seminars, webinars related to SEM and TM, the imaging, we'll discuss this more. So the SEM, like elemental mapping, is another thing we use fairly common. Use the SEM EDX of the area to provide mapping information. It's not an optical image or image. Keep that in mind. It's a elemental mapping based on different elements. So this one is a sugar sphere, which is inside a drug capsule. If you open up, there's a little beads inside it. The beads core usually is sugar-based, so we call it sugar spheres. Here is the control release formula. The coding for the control release is silica-based, and the active pharmaceutical ingredient is magnesium-based. So as the image you can see here, and we have a cross section, and you can get silica. Elemental mapping is all on the core. There's nothing in the middle, which makes sense because the coating is outside. And once you open it up, you see the magnesium is located at the high concentration magnesium are all inside the core. And the bottom is the EDX spectrum, and that's the one we actually want the most. Is just an example of a EDX spectrum look like. That will be something we use more for particular analysis. SEM EDX, of course, has strengths and limitations, just like any other technique. The strengths of it is it's really fast, pretty much like a minimum sample prep to no sample prep. And imaging resolution can go down to single nanometers. And EDX provide elemental identification. And of course, you want mapping, we can get mapping information as well. And then the limitation is if you have a very small particle, it may be consumed during the analysis because a high energy E-beam that hits the sample may consume the sample. And the samples must be vacuum compatible because that SEM chamber is a high vacuum chamber. The carbon tape sometimes we use to secure the particulates that may introduce contamination. The adhesive is very hard to get rid of once it's attached to the sample. So if something could be organic, you always want to start with FTIR first before going into EDX due to the carbon tape. And for non-conductive samples, we may have to apply certain coatings for example, like a silver or gold or platinum coatings to provide conductivity. That will introduce some elemental contamination as well. Now we had a brief introduction of the techniques, and let me give you a quick like a case study. So this case study here is a true story. However, we have regenerated spectral data using our internal samples, so I can present this demo for you guys. So the whole story happened was like I have a long-term client called me. He's like, Lynn, I have this rejected parts, rejected device coming back from the field. They said they observed some particles there, and it's about like a 4 millimeter length, and it's a plastic looking. It sent me a picture. It's about like 4 millimeter in length. And I was like, okay, so why do you think it's plastic? It does look quite like a plastic. And so usually when my client has some suspicions, I ask them why. And there's always a good reason most of the times. And he said that they have the only plastic parts they have on site, besides what's going onto their device, is a polyamide that could contribute to kind of shards like this. So I was like, okay, send me your reference material as well so we can have a comparison. So when we get the sample, we look at it, morphology, have a description of it. We examine the sample using an optical microscope. It's really sharp edged, actually, surprisingly. It's very stiff. It has a little off-white color and some transparent as well. So we agree with the client it's more organic looking. We should start with FTIR analysis first. And of course, we have a spectrum on top. This is for the 4 millimeter stiff sample. And the bottom is the reference client provided polyamide. And we think it's a relatively okay match, but there's something that's a little bit concerned is some characteristic band that's in the sample that's not in the reference material. One is around the 1,000 or the 1,100 range. We don't really see a significant amount here. And also the polyamide, the reference material has really two sharp bands. These two does not have a significant background. It's more intense than what's in the sample. Now there's a couple reasons that could contribute to it. First is they're not a good match. They're not exactly the same. Second one is the sample itself may be a mixture of something other than just the polyamide. It could be some other contamination lead to these spiky split of the peak as well, or shouldering. But to avoid that, we did is that we shaved the particulates a little bit to have a clean surface to analyze the sample again. The spectrum stays the same. So we're like, okay, we pretty much can rule out that this is a complex material with polyamide, but something coated on that. Could that be possible? This is not actually a good match. If it's not a good match, then what are they? So the story goes back to about a little while ago. Before that, we had a pharmaceutical manufacturer client that came to us. And they want to analyze every possible particulates contributing from a human body. So we got some body parts, not the big ones like hair, fingernails, and some dead skin, and we got some makeup could be falling off as well. That all contribute to their internal database. So one of it's actually Dr. Scott Graham's group took care of this project, and they come back. They're like, we think this was matching one of the samples our other client provided. It's more like a fingernail. So we're like, okay, interesting. So we went back and looked at that spectrum. It does look visually similar, but since it's our client data from a different client, we cannot share with our current one. So we went back to them. We told them our experience suggests that this could be a fingernail. It was a way of phone call. We told them like we think it could be a fingernail, and we consulted with them, asked them, do they want to proceed as a comparison? They said yes, for sure. They absolutely want to do that. Well, this is the case that we always want to check with the client. We don't do additional work until they approve. Sometimes like they may not to moving forward to get the actual information. So we consulted them. They're like, oh yeah, we absolutely want to find out what it is. Since it's a field rejected device, we want to figure out what it is. So we collect a reference sample from Dr. Graham. We logged in as reference number 2. And then we had a comparison of that and the fingernail. You can see the character band that's matching. Now it's showing some, and all the ones that showed up here as shouldering had the concerns. We had concerns before, now started showing up here. So this is the case we would consider it's a good match. We went back, tell them we think this is consistent with a fingernail. And a fun part is they actually went back to their client and said, hey, you guys reject this part on the field, but we don't think the contamination coming from our side. We're GMP facility, everything in the clean room. But the particles we observed seems to be consistent with a fingernail. Is there anything could happen on your end? Client actually on their field site, there was actually incident happened that day, was logged in, could contribute to the particles as a fingerprint. So they settled the case because the contamination are most likely coming from the field site. So that was a happy ending for that project. It not always be a happy ending for all particular analysis. There's certainly a lot of challenges associated with a particular analysis. First is that we have limited sample size, limited samples. The size and the number of the particles are very important. If you only have one particle, you have to pick and choose which one to go first, especially they're small. And of course, the location and isolation of the specific particles are very hard. For example, if we have a filter coming from the USP 788 method, we filtered out, you may have hundreds of filters of particles on that filter. On the optical image you get, they may be labeled to associate with a number. We have client coming in, I have one client saying, I want the particle number 137 analyzed for chemical identification. The numbers are not labeled on my physical filter. So we have to go in to rotate a filter under a microscope to line up with the picture to find out exactly which one is a 137 and isolate, extract that particular particulates for the chemical analysis. And of course, the contamination is a big concern for particles prior to analysis. We get environmental contamination and sample handling contamination as well. I always tell my client, if you don't have to touch that particle, don't touch it. Let us do the work. And so also sample matrix interference is always a big part, a big contribution to unwanted peaks. We have buffers and solvents in that. That's why after particle collection, if we can rinse it, we always want to rinse it to remove any buffer or matrix effect that particle is soaked in, especially when in solution. We have a lot of sharp tools in our lab for design for particle isolations as well. I have a little examples here. You may not even be able to see the tip, how fine they are. Of course, there are other techniques used for particular analysis. Sometimes FTR and SEM-EDS just not enough. We have Raman spectroscopy. It's fairly commonly used for FTIR as well. Sorry, for particular analysis as well, especially like carbon black, and sometimes even crystalline materials, polymers are pretty good for Raman spectroscopy. They're good candidates. And pyrolysis GCMS, especially for polymers, we want to confirm polymers. Sometimes you want to find out what other things, additives could be there as well. Pyrolysis GC will give more detailed information, but it requires a little bit more sample than just as for FTIR. And we have other ones like TOF SIMS, ICP, and ion chromatography if we want detailed elemental information. And of course, LCMS, GCMS, if we can put in solution, then those are really good organic determination, interpretation techniques. And a lot more, as we showed in that smart chart. Once we have the chemical composition determined, next following is always risk assessment, especially for the industry's potential patient risk is of concern. The size of it, the shape of the particles, especially if the sharp edge that could cut the inner organs, those are all of concern of risk assessment, of course. And the contacting method of human body, is it just dermal contact, or is it embedded or like implanting devices? And also contacting time with human body and also is of concern. and the chemical identity, of course, and the quantity of how much the particulates are there. And these are very specific to pharmaceutical and medical device industries. Some other industries you might consider like what the material is and how it affects the device or the parts. It will be of concern. But once we have the compositions of the particulates, so we also try to think about is how do we reduce them, since they're unwanted particles, we don't want them. And the source determination would be next step. So if you have a facility, so does it have a clean, is it in the clean area, is it a clean room? If it's a clean room, is it contributing from air filtration or not? The filters sometimes could be a source of contamination. So personal contact, human body, like I said, PDMS is in our makeups and hair gel, and our hair by itself and dead skin will contribute to particulates sometimes. And the raw material itself, some of them like maybe like rubber, they're sticky, maybe particles on them. They may not show up until you put that rubber stopper on some bottles, and when the solution rinse it come off. And you may have bags that's static, and they trap fibers and bring into your facility. And these you may have ways to remove them, like maybe rinse the part before using or transferring into your clean room. And some of them we see are byproduct. Your raw materials are fine as is, but when two things meet each other, they may not be compatible. When they're incompatible, two could be reaction products as well. Once we limit the source, what they are, and our next part is removal and reduction of the source. My team can help you figure out either we do a rinse or what's the necessary step to avoid the further contamination of these particulates. Why choose EAG? Like maybe some labs out there can do particulate analysis as well. Like you may ask, then why do I come to you? There's so many different reasons about. I can tell you all day long how good we are. We're way better than everybody else out there. But I think the most important to stay right on top is like the client confidentiality is the core to our business. We are a global leader for materials testing services with a broad range of instruments and expertise. That shows out on the smart chart, and also all the people in lab. So we can take on like even the most challenging materials and engineering related issues you have. And finally, so in highlight is the pandemic here right now that's affecting all of us. I want to stress that EAG Laboratories are committing to the safety of our staff and clients. Worldwide, we have complied with the latest COVID-19 regulations from state and local government, as well as CDC. Currently, all of our locations throughout the world remains open again, but we may have limitations on customer visits and sample collection sometimes, as this is a fluid situation where we have operating a laboratory. We're operating a laboratory at reduced capacity. We can also transfer your samples to another EAG location if it's needed, or they have faster turnaround time. We encourage you to discuss your options with our sales representatives.